class. I'm not talking about our review session here. So last time we talked about photosynthesis, and you know the goal of photosynthesis is for plants and other photosynthetic organisms to make their own food, namely glucose. And in doing photosynthesis, there are two steps, right? The first step is what we call the light-dependent reaction, well, because we need light for that. And the other one is called the Kelvin cycle, or the Kelvin-Benson cycle sometimes. Some people used to call it the dark reaction as well, but you know, it doesn't just take place in the dark, so it's a bit misleading. Anyway, there are two steps. First one is the light-dependent reaction. The second one is the Kelvin cycle. So we're going to review the light-dependent reaction right now, and we're going to cover a few more slides, and then we're going to talk about the Kelvin cycle. Now, as I said, the goal is to make glucose. Glucose is not made during the light-dependent reaction, not even a tiny little bit. Glucose is made in the Kelvin cycle. To make glucose, we need carbon dioxide. We already discussed the carbon dioxide usually comes from the atmosphere, and also, of course, is a byproduct of cellular respiration. So in the second step, during the Kelvin cycle, carbon dioxide gets incorporated and is going to be combined to another molecule that's going to be changed and changed again to eventually make glucose. Now, you know that making glucose during photosynthesis is an endergonic reaction. That means we require extra energy. Now, this is where the first step comes in, the light-dependent reaction. So the light, as you know, is the initial source of energy for photosynthesis. However, the useful source of energy for cells are molecules like ATP, and also in this case for photosynthesis, NADPH. So for the Kelvin cycle, make glucose, what we need is ATP, and what we need is NADPH. So the light-dependent reaction, the whole purpose of this is to make ATP right here and NADPH right there. Okay, so the whole point of this here is to make ATP and NADPH. So that's then the energy source we can use in the next step in the Kelvin cycle to make glucose. All right, that's important to keep that in mind. Now, first of all, the light-dependent reaction takes place at the thylakoid discs here. You can see this is the thylakoid membrane. Inside is the lumen of the thylakoid, and then this would be the stroma. The Kelvin cycle takes place in the stroma. Okay. Now, at this thylakoid membrane, we have a series of proteins. Okay. This is the Altron transport chain, very similar to the Altron transport chain that we've discussed in cellular respiration in the last step, which is oxidative phosphorylation. Just like in oxidative phosphorylation, what we have is we have here these proteins. There's one here, there's another one. Many of them are transmembrane proteins, and some, like this one here, can pump hydrogen ions into the lumen. And then we have ATP synthase. This is where we're making ATP. And just like in oxidative phosphorylation or cellular respiration, what happens basically is that electrons are moved from one to the other. It's going to energize some of these proteins, which are pumped. This transmembrane protein here is a pump. It can then pump hydrogen ion from the extracellular, from the stroma into the lumen of the thylakoid. So now we're going to have more and more hydrogen ions, and this gradient, we have more hydrogen ions inside the lumen of the thylakoid compared to the stroma. This gradient means that hydrogen ions now can move from high concentration inside the lumen to low concentration in the stroma. So, but as you know, hydrogen ions are ions, and ions don't just diffuse through the membrane. They need special proteins. Well, there is a special protein. It's ATP synthase. And so hydrogen ions are going to move through ATP synthase, and this movement of hydrogen ions gives ATP synthase to get energy to make ATP. Okay, so very similar, right? As we made ATP during oxidative phosphorylation in cell respiration. It's a little different, of course, because all this is driven by light, by photons that are hitting two places: photosystem 2 and photosystem 1. All right, so let's go through this one more time. So here are the different proteins in the Altron transport chain. The first one here is photosystem 2. Again, it's called Photosystem 2 because it was discovered second. And this is Photosystem 1 here. So it's kind of second in terms of events that are happening. Then we have here cytochrome oxidase. This is a transmembrane protein which can act as a pump to pump hydrogen ions. And then we have here ATP synthase. This is where we're making ATP, and this is where hydrogen ions can move from high to low concentration. And then here we also have NADP reductase. This is the enzyme where NADP plus is reduced to make NADPH. Again, the goal is to make ATP and NADPH. So what happens? We're starting out at photosystem 2. So now what we have is we have light striking photosystem 2. And in here are chlorophylls, and they can accept some of the electrons, and the electrons are going to be moved. Now what happens is in the presence of light, H2O water will be split to form oxygen. This is called photolysis. So the splitting of water is called photolysis. So what we're getting here is now when we're making oxygen, right? This is the product of photosynthesis, right? If you look at the net equation, during photosynthesis, oxygen is produced. Well, this is where it happens. Now, the other thing that happens is when you're splitting water, some hydrogen ions are dissociating. So we have more hydrogen ions that are accumulating here in the lumen. Second, we're losing electrons. Okay, as water is split, some electrons are lost, and these electrons are then accepted here by photosystem 2. And when light strikes them, these electrons get energized. Okay, so they increase their energy level. And that allows them that the electrons are then shoveled from one place to the other. Now, as the electrons now arrive here at this protein complex, this is a transmembrane protein, and it's a pump. It energizes this pump, and this energy from the electron allows this pump to move hydrogen from the stroma, from here, into the lumen. So now, as I said, we're getting more and more hydrogen ions. Number one, we get hydrogen ions because we're pumping them from the stroma into the lumen. So we get more hydrogen ions in the lumen. We're also getting some hydrogen ions by splitting the water here. So we get more and more hydrogen ions here inside the lumen. Now we have a hydrogen ion concentration, or an electrochemical gradient. This equation. So okay, we have a hydrogen ion gradient, and this hydrogen ion gradient allows us to make ATP indirectly. As you know, we have a movement always from high concentration to low concentration, as I said. However, ions can diffuse through. These ions, in this case hydrogen ions, they have to go to special channel proteins like ATP synthase. And this movement of hydrogen ions from high concentration inside the lumen to low concentration in the stroma gives ATP synthase to make ATP. ATP, of course, is made by ADP, and then we're adding an inorganic phosphate and we're producing ATP. So this is how we produce ATP. We also want to make NADPH. So now this is where it continues. So the electrons are going to continue being moved over to photosystem 1. Now, as these electrons are moving, they're losing energy on the way. Okay, and some of the energy is already used here to pump hydrogen ions. So they're losing some energy here. But now we have photosystem 1, and photosystem 1 also has chlorophyll pigments, and at P700. And once the light hits it, the electrons are arriving here, they get energized again. And so now the electrons get moved over, and they get moved to NADP reductase, to this enzyme. And this gives it energy to produce NADPH. In fact, the electrons are arriving here, they'll be accepted by NADP+. So when NADP+ receives an electron, it gets reduced to NADPH. And so this movement of electrons, and so finally electron is going to be accepted by NADP+, will reduce it to make NADPH. So this is how we make NADPH. So again, the goal were two things. Number one, make ATP, and number 2, make NADPH. And in both cases, all this is energized by the light and the electrons moving. So again, first light hitting here, then here electrons arriving here, gives it energy. We're pumping hydrogen ions into the lumen. We get a hydrogen ion gradient. Then hydrogen ions can move from high concentration to low concentration inside the lumen to the stroma through ATP synthesis enzyme, and we're producing ATP. Then electrons keep moving, get re-energized here, and eventually here at NADP reductase, the electron are accepted by NADP+, and we're making NADPH. Okay. Again, this was the whole goal. The whole purpose of light-dependent reaction is make ATP and NADPH because we need to use those two to make glucose. Okay. Without NADPH and ATP, we cannot make glucose. So that's the whole point of that. So now I'm going to go through the next couple of slides. Most of them are basically showing the same thing that I just explained already. So I'm going to be kind of repeating myself a little bit, but it just shows a slightly different. Now this one here shows kind of a zigzag shape of the energy of the electrons. So the idea is this is photosystem 2, and then this is photosystem 1. Now electrons are arriving here at photosystem 2, and then these electrons are energized by light, by photons. Now the energy of the electrons increases, goes up. Then the electrons are moved to other places, cytochrome complex, for example, and some of the energy is used to pump hydrogen. So as they're being moved, and as the energy is used to pump hydrogen, they're losing some electrons. So the energy gets less and less. And then they're arriving at photosystem 1, again, light strikes, and then the energy becomes more, and then they're gonna get moved over to NADPH, dehydrogenase, another enzyme here, and we're losing again some electrons. So it's kind of like a zigzag curve here. So you can see a light strike, energy of the electron becomes more, they get moved, electron becomes less, light strikes again, energy becomes more, they get moved, and we're losing some energy again. All right, so that's the idea of that. All right, so again, don't worry about all the details about photosystem 2, but again, the whole thing started at photosystem 2. Let me just go back one more time to this slide, sorry. Okay, this is photosystem 2 here, right? So we're going to be talking about this here first, and then we're going to talk about what happens at photosystem 1, okay? All right, photosystem 2, as we discussed earlier, photosystem 2 has P680. P680 are these chlorophylls, pigments, and they're called P680 because they absorb best at wavelength 680 nanometers. And so they form a complex which is called an antenna. And so here, basically some of the electrons are being energized at this place. At the same time, at photosystem 2, we have photolysis, right? So water splits. When water splits is, number 1, we're forming oxygen. Number 2, some hydrogen ions are dissociated, so we get more hydrogen ions inside the lumen. And then we have the electrons. And these electrons then can be used, and they're going to be energized when light strikes photosystem 2. Now, the whole point of photosystem 2 is this here. We want to produce ATP, or photosystem 2, right? Just going back here for a moment. So photosystem 2 is indirectly responsible to produce ATP, right? I explained that, you know. So electrons are energized. They're going to be moved over in cytochrome C. We're pumping hydrogen ions in here, and then this hydrogen ion gradient can be used to produce ATP. So ATP production is indirectly associated with photosystem 2. On the other hand, producing NADPH is more closely related to photosystem 1, right? Because light strikes the electrons, they get moved over here at photosystem 1, and then we can make NADPH, okay? So when you think about ATP production, that's more related to photosystem 1, and making NADPH is more related to photosystem 1. Sorry, this is 1 and this is 2, right? So, all right. Okay, so again, indirectly, photosystem 2 is used. The electrons are excited, then are moved along the electron transport chain in the thylakoid membrane, and we then arriving at this one enzyme, this one protein, transmembrane protein, which acts as a pump, and it pumps hydrogen ions into the lumen, and we're producing a hydrogen electrochemical gradient. And this gradient can be used to produce ATP because the hydrogen ions are moving from the lumen of the thylakoid into the stroma through ATP synthase, and we're producing ATP. So as I said, photosystem 2 is indirectly related to ATP synthesis. Again, this explain this whole process here one more time. We have photosystem 2. In photosystem 2, electrons are excited. They are moved on to this particular enzyme here. This is a pump, and this pump can move hydrogen ions from the stroma into the lumen. This is the other way around in the other picture, right? Looks like this one here is the stroma, and this is the thylakoid lumen. Okay, so we're pumping hydrogen ion into this into the lumen of the thylakoid. We're getting a hydrogen ion concentration gradient, and then therefore we are then hydrogen ions moving from high concentration to low concentration through ATP synthase, and we're making ATP. Remember, this is very similar to oxidative phosphorylation, which we had in cell respiration. Now, only here, we don't call it oxidative phosphorylation, we call it photophosphorylation. Photo means light. The reason why we call it photophosphorylation, right, because the electrons have to be moved and they are energized originally by the light hitting it. So this photophosphorylation is very similar to oxidative phosphorylation, only that the energy we're getting to move the electrons to this protein, to this pump here, to make this gradient and then to make ATP uses light. So it's called photophosphorylation, this process. They are the same because you're talking, I assume they are the same because it produces energy. You mean oxidative phosphorylation is the same as this one because in both cases we're producing energy? I would change it slightly. I wouldn't say they both produce energy. You know, think about at the beginning we said we can't really make energy, right? We can only change energy from one form to another. But they are the same that in both cases we're producing ATP. And they're producing ATP somewhat in the same fashion, right? Oxidative phosphorylation, we're producing ATP because we have a hydrogen ion concentration, and ATP moves through ATP synthase. And that's the same way here, isn't it, right? We have a hydrogen ion gradient. We have more hydrogen ions in the lumen than in the stroma. Again, we have a hydrogen ion gradient also in oxidative phosphorylation. And hydrogen ions move from high concentration to low concentration to ATP synthase. That's the same again, oxidative phosphorylation and photophosphorylation, and then we're making ATP. Yeah, so they're similar in the way they make ATP, exactly. The only difference, of course, the original energy comes from the light here, right? Because the original energy, we need energy to move the electrons so that the electrons are arriving here at this pump. All right. Now, you can read through this here one more time. The only other additional information here, this is basically the whole text here, what happens at each step here. So I don't want to read through it. But basically, again, we're talking photosystem 2. We have photolysis, water is splitting, we're producing oxygen, electrons are released, and lights are hitting photosystem 2. Now, they're hitting the light harvesting complex. Okay, this is what absorbs the light energy, and that can be then used to energize the electrons to move them on. And so again, this is the same thing that I just explained here. We then producing a hydrogen ion gradient because there's a hydrogen pump. We're pumping hydrogen into the lumen, and then this gradient can be used, this electrochemical gradient can be then used to make ATP using ATP synthase in a form we call photophosphorylation, right? Not oxidative phosphorylation, but photophosphorylation. So again, you might want to read through this here if you didn't get it right now, but this is an important part as well. All right. Now at photosystem 1, again, photosystem 1 is indirectly responsible to produce NADPH. So again, this is photosystem 1 right here, right? So photosystem 2 was right here. This is photosystem 1. Photosystem 1 receives the electrons that have been shoveled, but the electrons have low energy. Now, the photons are hitting, the electrons are excited again, they gain energy, they're going to be moved over to this complex here, NADPH dehydrogenase, and then the electrons are received by NADP+, and then NADP+ is reduced to NADPH, and again, we're making NADPH. So photosystem 1 indirectly is used to make NADPH because NADP+ accepts the final electrons here. So photosystem 1 is not really related to making ATP, right? That's related to photosystem 2. Okay, so again, this explains it here again. This is for photosystem 1. We have a situation where light strikes, electrons are energized, and the energy is then indirectly used. Well, not indirectly. The electrons are moved, and then the electrons are reached by NADP+, and we're making NADPH here. And for each, in this case here, basically getting 2 electrons are being accepted here. We have, in this case, NADPH. Yeah? So again, photosystem 1 is indirectly responsible to make NADPH in this case. All right, this one more time, right? Just quickly. So we have here photosystem 2. Photosystem 2 gets energized here by light hitting photosystem 2. The electrons are energized and get moved over from one place to the other. At the same time, we have photolysis. So water is split, making oxygen. Hydrogen ions are released, and electrons. The electrons are moving. They're energized. They're moved over to the pump. We're pumping hydrogen ions, and the hydrogen ions pump then indirectly is responsible to make ATP. The electrons are moved over to photosystem 1. They're losing some electrons, but they're re-energized by light hitting it. Electrons are moved on to NADP+, and making NADPH. And again, ATP and NADPH can be then used in the Kelvin cycle in the next step we're going to discuss in just one moment. All right, quick summary of all this here. So during this light-dependent reaction, a couple of things happen. Number one, we're producing oxygen, right, doing photolysis. We're also producing then hydrogen ions and electrons that can be used in the next step. And we're producing ATP, right? ATP is produced because of the hydrogen ion electrochemical gradient that's used. Number one, we get hydrogen ions in the lumen by splitting water, and we're also getting more hydrogen ions into the lumen of the thylakoid because they are pumped from the stroma that we are making here. And then at the same time, this is this happens mostly at photosystem 2, and then photosystem 1, NADPH here is produced. Electrons are received here. All right. Now I have to mention one more thing here before we talk about the Kelvin cycle. Now, what I explained to you right now, we call it the non-cyclic cycle, okay? The non-cyclic pathway of the light-dependent reaction. As you know, in this process here, we're producing both ATP, we're producing both ATP and NADPH. But sometimes we need to produce more ATP than NADPH. And there is a way to shutting down making NAD. We don't need to make more NADPH than we need. So there's a way to shutting down the production of NADPH. And this is by what we call the cyclic photophosphorylation. So in a cyclic cycle, let me just go to the next part here. What happens this here? This is photosystem 1. You know, normally at photosystem 1, let me go back here to the original slide here. Okay, normally this is photosystem 1 right here, okay? So normally what happens at photosystem 1, electrons are moved over and then they're received by NADP+. And then we're making NADPH. So this is called the non-cyclic cycle. In the non-cyclic cycle, electrons are moved on to NADP+, we're making NADPH. Now, if we don't want to make much NADPH, we have a cyclic cycle. In the cyclic cycle, the Ultrons are not moved to NADP+. They are just basically bounced back, okay? So if the Ultrons are not moved to NADP+, we cannot make NADPH, okay? So it's a way to stopping making more NADPH. That's the cyclic cycle. Let's go back here. There you go. There we go. So this is here photosystem 1. Normally, Ultrons are excited, and these Ultrons are going to be moved on to this enzyme, and they are received by NADP+. But in the cyclic pathway, they're just going to go back again. So in a cyclic cycle, we do not move the Ultrons to NADP+, and therefore no NADPH is made. Okay? So normally, if we have a non-cyclic cycle, we're going to have equal number of ATP and equal number of NADPH is made. But sometimes we want to make more ATP than NADPH. If we want to make more ATP than NADPH, we can stop making more NADPH by going to a cyclic cycle where the Ultrons are not moved on to NADP+. Now, and some bacteria, some algae, for example, they tend to do it all the time. All right. Let's talk about the Kelvin cycle. So in the Kelvin cycle, one more time, this is the goal to make glucose. So to make glucose, we need to use carbon dioxide, and we need to use ATP and NADPH that were produced during the light-dependent reaction. Now you notice it's not an equal number that we need. Right, so usually to make 1 glucose, to incorporate 6 carbon dioxide, we need 18 ATP and 12 NADPH. Right, so we need 50% more ATP than NADPH here in this case. Right, so we need more ATP, and of course we need 6 carbon dioxide. Right, glucose is C6H12O6 right up here. There you go, 6 carbons. Where do we get the 6 carbons from? Well, from 6 carbon dioxides. And so they have to enter the Kelvin cycle. All right, let me just go through the Kelvin cycle here a couple of times, just quickly. Let me just see if I can zoom in a little bit so you can all see it. All right, so first of all, again, the Kelvin cycle. Right. I mean, the point is, the normal non-cyclic pathway, the normal light-dependent reaction is the non-cyclic pathway. In the non-cyclic pathway, we make an equal number of NADPH and ATP. Now, I just told you to make glucose, we actually need to make more ATP than NADPH. So it would be a waste of energy to make more NADPH, right? So we can shut down, we can stop making NADPH by going to the cyclic cycle, okay? When if you go to the cyclic cycle, the Ultrons are not moved on to NADP+, and we're not making NADPH. All right, here's a Kelvin cycle. So Kelvin cycle, it happens all inside the stroma. So carbon dioxide enters the stroma. Here we have carbon dioxide, 1 carbon dioxide at a time, but total of 6. Now, the carbon dioxide is going to be added to ribulose bisphosphate. Ribulose bisphosphate has 5 carbons. This has 1 carbon plus 5 carbon. We're making something that has 6 carbons. The 6 carbon molecule is then split into 3 carbon molecules, to 2 3 carbon molecules. That is going to be changed to another 3 carbon molecule over here. And changing this takes ATP and NADPH, right? So we have to change this around. And then these 3 carbon molecules can be put together to make glucose. Glucose had 6 carbons, so 3 carbon molecule and another 3 carbon molecule, we can make glucose. And then we're gonna have some of them left over, I'll explain in just a moment, and we can go back to the original one, this is ribulose bisphosphate, and we can start a new cycle. So the cycle has to enter 6 carbon dioxide to make 1 glucose. Let me go to the next slides to explain that step by step. Well, maybe go a little bit higher. All right, there we go. We're starting out with carbon dioxide. Carbon dioxide, of course, is 1 carbon. we adding the carbon dioxide. Over here, let's have a look at this one. This is carbon dioxide. We're adding carbon dioxide to this 5-carbon molecule. This 5-carbon molecule is called ribulose bisphosphate, or RUBP. Okay, so we're adding carbon dioxide to this 5-carbon molecule. Now we're making a 6-carbon molecule. You don't have to know the name of it. Now this 6-carbon molecule, there it is, gets split into 2. So it gets split into 2 here. So we're splitting this one here into 3-carbon molecules, right? So we go over here, we make this one here, and this gets split into 2 3-carbon molecules. Now this 3-carbon molecule is called PGA, or phosphoglycerate. Okay, so we're making 2 phosphoglycerates, 2 PGAs. The 2 PGAs are then changed to 2-PGEL, that's phosphoglyceraldehyde. And the phosphoglyceraldehyde can then be used to make glucose. Now, I said 2, but in reality, we're starting out with 6 carbon dioxides. We're going to add them to 6 ribulose bisphosphate. So we're making 6 molecules that have 6 carbon molecules. 6 carbon molecules. Those 6 carbon molecules get split into 2. So therefore, we're going to have 12 3-carbon molecules. The 12th is called PGA. So we have 12 PGAs. Those 12 PGAs that have 3 carbons gets again changed to 12 PGEL, okay, phosphoglycerol. So we have phosphoglycerate gets changed to phosphoglyceraldehyde. So 3 carbon molecules, 12, we have 12 phosphoglyceraldehydes. And now, in the end, let's just go over here, we made 12 PGL, 12 phosphoglyceraldehydes. The phosphoglyceraldehydes, let me just go down here. The phosphoglyceraldehydes are 3 carbon molecules. These 3 carbon molecules, we can take 2 of them together, put them together, and making glucose. So we only need 2 phosphoglyceraldehydes. We only need 2 PGALs to make 1 glucose. We made 12 of them. We used 2 of them to make glucose. So we have 10 PGALs left over. And these 10 phosphoglyceraldehydes, PGALs, can be used to make, again, the starting compound, which is ribulose bisphosphate. And the ribulose bisphosphate can be used in another cycle to accept carbon again. We make the 6 carbon molecule. The 6 carbon molecule gets split into 3. We have PGAs, and then the PGA gets changed into PGEL. So that's basically how this process works. Let me just go back one more time. Let me do it one more time. Okay, we have 3 minutes left here. All right. So in order to make glucose, we're going to be using 6 carbon dioxides. Okay, so 6 carbon dioxide can be used to make 1 glucose. So we're going to have 6 carbon dioxides going to be entering the cycle. The 6 carbon dioxide, each carbon dioxide is going to be attached to one of 6 5-carbon molecules, right? It's called ribulose bisphosphate. You can see it has 5 carbons. So we're making a 6-carbon compound. And because we use 6 carbon dioxide and 6 ribulose bisphosphate, we can make 6 carbon molecules. In the next step, these 6 carbon molecules get split into 2 that have 3 carbons. So these, now, because we have 6 in total here, we're going to split 6 into 2. We're going to have 12, right? So we're going to have 12 3-carbon molecules. Those 3 carbon molecules are called PGA, or phosphoglycerate. The phosphoglycerate then in the next step gets changed to PGEL, phosphoglyceraldehyde. So we're going to have 12 phosphoglyceraldehyde. They all have 3 carbons. Again, we want to make glucose. Glucose has 6 carbons. So we can put always 2 of the PGALs, 2 phosphoglyceraldehyde, can be joined together to make 1 glucose. But we made 12. And because we made 12, we only use 2 to make 1 glucose. 10 of these PGLs can be used again to make ribulose bisphosphate, which is a starting molecule in the Kelvin cycle. And so in the end, we made 1 glucose using 6 carbon dioxide. At the same time, you can see here, we use ATP and NADPH. So in this case, you can see we're using ATP is used up to change it here, and we're also changing NADPH here in the next step here. Now, NADPH is used twice, okay? So we're changing PGA to PGAL. We need 12 ATP and 12 NADPH to change it. Then the PGAL is going to be joined together to make glucose. Now we have 10 PGA left over, and we have to change it back to ribulose bisphosphate. Changing it back to ribulose bisphosphate takes another 6 ATP. So we need to use another 6 ATP to change it back to this ribulose bisphosphate, which is a starting point. And that's why we're making it. We need a total of 18 ATPs here for a cycle, for these cycles here of Kelvin in the Kelvin cycle here, to make 1 glucose. All right, we're done with this lecture. I'm going to stop recording in just a moment. Do you have any questions about this here? And by the way, we're finished with the chapter 2, right? So we have one more slide. This is just the same slide here as the first one here. So this is the end of the chapter, okay? So again, we make glucose. That was the whole point. Photosynthesis about making glucose. But remember, in the first step in the light-dependent reaction, first we have to make ATP and NADPH. Right? ATP and NADPH are the direct energy source to make glucose. The light only indirectly, right? We use the light indirectly to make ATP and NADPH, and then the ATP and NADPH were then used in the Kelvin cycle to make glucose. And so the glucose, of course, can be used later on in cell respiration to make more ATP, or the glucose in plants, for example, can be stored as starch and can be used later on as an energy source. There is a bit of information. How many chapters are left? So we finished chapter 8 now. We'll decide about the quiz here. I'll give you another vote here. It was very close. I don't think we finished the vote here. I'll let you vote online somehow, okay? I'll set up later today or tomorrow. How many chapters are left? We have 3 more chapters left, I think. Just a second. Yes, we have, so 8, we have 9, 11, and 16 left over. So 3 more chapters, okay? So 3 more weeks. The thing is this here. So we're going to cover 1 chapter per week. Don't be too worried. Those chapters are going to be faster, okay? There's not as much information in there. And in fact, we got a meeting before we started the semester, and we threw out a lot of information in the remaining chapters. So we will be able to cover them easily, okay? All right, let me stop the recording. And if you have more questions, I'm staying around.